ways to integrate methods for metabolic engineering from automated tools and manual curation. To set the stage for this talk, I'm going to start, like a lot of talks start, with the DNA sequencing sort of paradigm, which is that sequencing information is increasing exponentially. You may notice on this axis right here, this is a semi-log plot. So what we're seeing here is that the number of complete genomes is growing very rapidly as we develop new technologies like next-generation sequencing. So there's a tremendous amount of sequencing information. So what can we do with that sort of information? Well, one thing that we can do, and that we do a lot through labs like the Price Lab in systems biology, is we make those genome sequences into functional networks. And specifically, what I've done during my work is work with metabolic reconstructions. And what I mean by that is I take a genome, so this is just a fragment of a genome here, and I can take those genes and I can annotate them, which involves first translating them into the proteins that they code for. So in this case, these three proteins I've shown come together to form a protein complex. And then those protein complexes or proteins are enzymes that catalyze chemical reactions. And by annotating my genes in this way, I end up with this reaction pathway here, where the different genes are attached to the reactions that they are linked to by catalysis. By putting together the entirety of the reactions across the entire genome, I come up with what I've shown down here in the right-hand corner. Now, this may look a little bit like a blob, but what's actually in here is a whole bunch of these different reaction pathways, but it's over the entire genome. So I'm talking about something on the order of 500 to in the thousands of reactions, depending on the system. Now, over the last about 15 years, since 1999, the systems biology community has constructed metabolic reconstructions for a wide range of different organisms across the tree of life. A lot of this work has been up here in proteobacteria, but there's been a fair amount of exploration across other branches of the tree of life. But there are a fair number of phyla that we haven't yet characterized. So even though the scope of models is growing, there's still quite a bit of work to do. Now, why are these metabolic reconstructions actually important? Well, they tie into a number of different processes in the biochemical and biomedical spheres. And specifically today, I want to focus more on industrial biotechnology. And what I'm really talking about doing there is developing methods for metabolic engineering. How I can take an organism and take all of its metabolism, all of the biochemical processes that that organism has, and I can engineer them. I can alter them to do what I want them to do, to take in some raw material and produce an important industrial chemical, like a biofuel. Now, in both of these cases, these different, both DNA sequencing and metabolic reconstructions, putting together the sequences or putting together the models that we've made requires a lot of automation. And when we use automation, we get some really great gains in speed. DNA sequencing, when we've progressed to next generation technologies, has increased the speed dramatically and lowered the cost as well. But what we run into with DNA sequencing is that instead of these larger reads with a lot of overlap that are easier to sort of link together when we get them, what we get from next generation sequencing is a lot of smaller reads. And by doing this in a massively parallel sort of way, we can do it a lot faster than we do these longer reads. But these pieces right here are a lot more difficult to put together than are these pieces on the left when we don't have some sort of template. What we would call de novo sequencing. So we're opening ourselves up to some errors in how we're putting those sequences together. And similarly, when we talk about metabolic reconstructions, we're opened up to errors where if I feed my genome into some database to annotate it, I might pull out an incorrect annotation like this one, or the annotation might actually be missing. I might have no information in the database about what this particular gene does. And this can present somewhat of a problem because as we're looking to do metabolic engineering, it's important that we learn about what organisms are actually doing, what metabolism actually looks like, and these errors make it difficult to discover what's actually going on. So to mitigate these errors, Really, the best ways that we have right now are to use more manually curated data. And what I mean by that in the case of DNA sequencing is instead of relying solely on the next generation reads, the small reads, I can map them to a reference genome, which is something that looks a lot like the organism that I'm going for. The genome has a lot of similarities, so it's sort of like taking a jigsaw puzzle and putting it together based on the picture that's in front, rather than just putting it together without any sort of template. Similarly, for metabolic reconstructions, instead of relying solely on automated annotations and what I can find in a database, I can actually take my genes and go back to literature, where somebody did a biochemical characterization of a particular gene or a particular enzyme. And I can use these methods to go back and refine what I get through the automated method and to correct these errors. And this really feeds into what I've focused on for the last 5 or so years. My central question in my thesis has been, how can we develop methods? How can I develop methods and resources that allow us to put together manual and automated methods to really get the best of both worlds? The speed of the automated methods, but the precision of manual methods. And more specifically, I'm going to be starting by talking a little bit about defined growth media and my work there. And then I'm going to move back into the space of metabolic reconstruction specifically. First, talking about my work putting together a model of my own through manual curation, and then talking about a method that I've developed to take a manual model and propagate it to a new organism. Let's dive right in. I'm going to start talking about microbial growth media. Now, this is a little bit different from what I've talked about in the introduction, but it's actually really closely linked in a number of ways. And the way that I want to link these two is by pointing out that allowing us to culture things in lab, when we can actually grow an organism in lab and study it, we can do biochemical characterizations to learn what's actually going on in that organism. We are able to do things without growing organisms in the lab. There's culture-independent methods. But growing things in the lab is the way that we best know how to characterize them. And unfortunately, if we look across all of the organisms that we know, particularly in the microbial sphere, the majority of bacterial and archaeal phyla cannot currently be cultured in a lab. The estimates range somewhere in the range from about 95 to 99.99% of microbes cannot currently be cultured. And partly this is just because there's a lot of diversity out there, kind of a mind-blowing amount of different organisms that we just haven't studied very well yet. And because I can't grow these things in lab, it's more difficult for me to learn about their metabolism and more difficult for me to learn how to make tools that can leverage those metabolisms in order to create industrial biotech processes. So what I really wanted to be able to do when I started this project was find some way, if I went into some environment that's very rich in microbial life, such as the soil environment, which has millions and millions of different types of microbes. If I go into one of these environments and I'm able to pull out sequencing information, which is something that I can get without culturing, or whatever information I can get from the environment about that organism, can I find some way to go into the lab and grow it? Is there some missing link here? Well, the problem that I found when I tried to address this problem, this link, is that there's really no training set. There's a lot of information in literature where microbiologists and biochemists have grown different organisms, but it's all rather disparate. It's in these separate literature sources. So what I decided to do was to bring these sources together, and I created a media database, and that media database is creatively called MediaDB. And it's a database for defined media. Now, defined has a special meaning here, so I'd like to define defined for you, if you will. When I say defined, what I mean is that all the chemicals in the media have known compositions. So a good example is sodium chloride. I know it's one atom of sodium, one atom of chloride. This is as opposed to complex media, which contain substances that we don't necessarily know the exact chemical composition of. One good example is yeast extract, where we have a decent idea of the sort of things that are in yeast extract, but we don't know exactly what's there. we do know that it tends to have a stimulatory effect on growth for a wide range of different organisms, so it's used a lot for growing them. Now, by putting together a database of defined media, what I've done is I've specifically outlined the chemical growth conditions for those organisms. So when I think about developing tools for metabolic engineering, I know exactly what the inputs to my system are, rather than having kind of a blurry idea of what's going into the process. Now, getting back to the database itself, this is basically what the database looks like. It's a series of linked tables of information with the arrows sort of pointing to different information and where it came from. And I'd like you to kind of direct your focus to this table here, the growth data table. What that table does is it brings together some medium represented by its identifier, medium ID, an organism strain represented by its strain ID, and then, of course, the literature source where that came from, so that I can actually track back to those literature sources, and I have a way to easily check everything that's in my database. And then, if there are other information that are associated with that growth condition, I have the ability to store the growth rate or the temperature or any additional information like that. And when I put this database together, I was working with a couple of undergraduates that I had with me in the lab, and together we were able to put over 700 different growth conditions in the database, which was a great amount of looking through literature, a couple hundred literature sources, I believe, something over 100, and basically just manually pulling out all these different growth conditions and formatting them correctly. And I realized that if I really wanted to be able to capture as much media information as possible, it was important to make this resource open to the public. So what I did is I worked with a software engineer in Seattle at ISB named Victor Casson. And together, we put together this website. And this is a page for the website. This is a particular medium composition, so it's got its name here, which may or may not mean something to you. But more importantly, I have the list of the 12 compounds that are part of that media and their amounts in units of millimolar. So I can immediately go to this media composition and see exactly what's in it. And I can also see what organisms grow in that medium, the source it came from, and the growth data record, which may have extra information like the growth rate or compounds that are secreted. So by putting this resource out there, I filled a need where there was no database like this when I started my work. This was the first database of its kind containing chemically defined media for different fully sequenced organisms. So now that I had all this information in one place, I decided to look over them and sort of see if there was any sort of trend I could find. And what I did was I looked at the most frequent compounds. Now, to sort of explain this a little bit, what I did for every organism in the database is I looked at every single growth condition. So if an organism had 10 different media, I grouped those media together and took all the compounds that occurred there. And then looking across all different organisms, I took counts of how many times a certain compound occurred over every organism. So for instance, calcium chloride here at the top occurred for 86% of organisms in the database. And really the trend that I found when I looked here, I didn't really find too much in the way of amino acids, and with carbon sources, I did find that glucose occurred almost half the time. It happens a lot in media. But I actually want to direct your attention more to this left hand, which are these compounds and their percentages right here. These 10 compounds, if you're familiar with growth media, if you aren't, that's okay. These are the sorts of things that you would often see in what you might call a trace mineral solution, a vitamin solution, or a general salt solution. These are the sorts of things that biologists who design cultural media use quite often. They're kind of generic solutions where the idea is that organisms have a certain set of compounds they require for growth and that they're similar between a lot of organisms. So if I throw most compounds that I know are necessary for growth at that organism, I expect that it can grow. It's not necessarily that every single compound in that solution is necessary, but it's sort of like broad spectrum antibiotics, where if I throw a lot at the problem, I'm assuming something's going to work. And what that sort of suggested to me as I was looking at these data was the idea that when we define media, it's not necessarily that we're doing it specifically to a certain organism. We certainly do try to tailor to some extent, but a lot of media look really similar. There's a certain uniformity. And to get a little deeper into that, what I did is I took these same data and I used them to cluster organisms. So this also takes a little explanation. Basically, what you're seeing on the left, this is a heat map where the areas of red are places where an organism is known to use a certain compound in its growth media. And black is where it's not. And more important than the heat map itself, actually, is this right here. This is the tree that I constructed, which basically shows the similarities in organisms based on the compounds in their growth media. So looking at this tree, really the thing that jumps out most at me is the fact that E. coli up here, actually, if you follow its branch, you'll find it actually clusters away from every single other thing in the database. Now, why is that? Well, what it means is that the media are least similar between E. coli and everything else. And the reason is because E. coli is basically the best studied microorganism on Earth. And because of that, we know how to grow it in a lot of different things. So we've used a whole bunch of different compounds here that we wouldn't necessarily see in any other organism. So E. coli looks unlike anything else we've seen based on its media. And this is very different from if we were to, say, cluster organisms based on phylogeny, which is basically a metric of how similar two organisms are. It sort of measures evolutionary distance. And if I do that tree, which is shown here on the right, I see a very specific clustering. These right here are basically the bacteria. Up here are the archaea. And then over here are the eukaryotes that are in the database, for the most part. So there's a very distinctive grouping there, but what I didn't really find was the same sort of distinctive grouping here on the left by organisms and their compounds. So what this suggested to me was very similar to what I talked about for the previous slide, which was that organisms aren't necessarily grown based on their phylogeny or how similar they are to one another. Growth media are largely determined based on what's worked for other organisms before. So to sum up this section of the talk, I found that ultimately my database filled a need where it just hadn't been filled before. There was no centralized repository of growth media to this point, and now there is. And it's got a number of conditions. It's open to the public so that more information can be contributed, and I've gotten some contributions of growth conditions since publishing in 2014. But looking across the media that are there, I don't really see a connection between phylogeny and media compounds. And in fact, it really suggests that a lot of media look very similar in regards to what compounds pop up most frequently. So moving along from this section, I want to get back into metabolic modeling, which is really what I started the talk with. And specifically, I mentioned that automated methods and manual methods need to be used together. And what I want to show you is a demonstration of using manual curation to supplement automated curation for building a model of Methanococcus maripolutus. This is what you might call the methanogen section of the talk. So to draw sort of analogy between what you might see in a factory, a cell can be used as sort of a microbial level factory, where basically what's coming in is growth media. These are the raw materials that are coming into my system. And then the cell performs a number of operations. Now, on a chemical plant, we might call these unit operations, where one chemical is converted to another and then separated. Well, a cell has internal metabolism, where every single one of these arrows is a biochemical reaction. It's a biochemical network of different reactions. And what these reactions do is, on one hand, they convert the growth medium to these secreted products. Now, this is what I might want from my chemical process. These are the things I'm trying to make. For the cell, these are really byproducts. And the reason is that what the cell tends to want to do is the cell wants to make more cell. It's a very common thing among cells. They like to be able to survive. They like to be able to grow. And so the cell is actually concerned with making these biomass components here, specifically ATP, which provides the energy for growth. So as I look at my cell, there's some similarities to a chemical plant, but I also have to consider the fact that my cells actually need to grow, which is not something that I have to worry about when I'm doing a purely chemical process. And a metabolic model can help me simulate this. And the way that it does this is if I construct my reaction network, I have something like on the left. Now, the way to read this, if you're not familiar with it, is I have three reactions, 1, 2, and 3. In reaction 1, I can see that metabolite A is reacting with metabolite B, and metabolite C and D are products of that reaction. Now this is a network sort of representation, and if I want to represent it mathematically, I can construct what's called a stoichiometric matrix. So basically, every single reaction is a column in this matrix, like reaction 1 right here, and I represent what's called the stoichiometry of the reaction, which is basically the coefficients of each metabolite. So metabolite A goes into reaction 1, it's a reactant, so it gets a -1. So does metabolite B. And metabolite C and D come out, they're products, so they get +1. And if I do this for every single reaction in the network, generally something in the range of 500 to 1,000 reactions in my experience, what I have is I have a genome scale model, and then I can simulate that model. And the way that I simulate it is I assume mass conservation. And what that means is that for metabolite D, for instance, I cannot use metabolite D for reaction 2 or reaction 3 unless it's first created by reaction 1. Everything that's created has to be used, everything that's used has to first be created. By making that assumption and simulating my model to maximize growth and adding a couple other constraints, I get what's called a flux distribution. Basically, I figure out where metabolites are going. I have a prediction of what's going on in the network. And what that looks like is shown here on the right, where I have a lot going through reaction 1. I'm making a lot of metabolites D and C. And then what I'm saying is that most of that goes through reaction 3. There's a lot of flux in this reaction, and there's not very much in this reaction as shown by the tiny little arrow that you may not be able to see from the back. So that's a flux distribution, and that's what my model can simulate. Now I haven't forgotten about the fact that my model needs to grow. And one of the consequences of that is that there are certain metabolites that I need to make. I mentioned, for instance, that ATP tends to be an essential metabolite. And for the purposes of this slide, I'm going to say that metabolite E right here is essential. If I don't make metabolite E, my cells die. For instance, if metabolite E for a human was skin cells, I need to be able to make skin cells so I have skin so that I can exist in this world. So, in order to make these biomass components, I need to have some sort of path that leads to them. And there's many cases where I might have a gap like this, where I know that something has to fill that gap. I know I need to make metabolite E, I just don't know how I do it. There's a couple ways to fill this gap. One way is through what I would call a maximum parsimony. It's the simplest path, the shortest path. And that would involve going to one more metabolite, in this case metabolite F, and then down to metabolite D. So just 2 more steps, maximum parsimony, very simple. The problem is that the simplest path is not always the most likely. For those of us who have driven cars around towns, we know that we oftentimes don't take the shortest path from one route to another. Instead, we take a path that's more efficient or one that just gets us there in a faster time, even if it's not the shortest route. Well, cells also don't necessarily follow the shortest path. And so what a previous student in the Price Lab did is he created an algorithm called probabilistic annotation. It's a maximum likelihood sort of approach. And that student was Matt Benedict, who did a lot of work with Dr. Metcalf. And what his algorithm does is if I feed my genome into a database, it calculates likelihoods for each reaction in that database. So for these reactions, for instance, the probability of this reaction being in the model is 0.9 on a scale of 0 to 1. So it's very likely that that reaction is part of my model. And by assigning these likelihoods, I can fill these gaps using the most likely reactions rather than just the shortest path. So putting this all together and kind of stepping back a step, I can look at my overall process of putting together a model. So what I do is I can feed my genome into an annotation database, in this case the K-base. It's the Department of Energy Systems Biology Knowledge Base, but we'll call it K-base for the rest of the presentation. And then I can gap fill that network to make sure it grows. Now after gap filling, I need to move up here. I need to refine. And I do that by going back to literature sources, like I mentioned at the start of the talk. And by doing that, I can find a lot of information, a lot of biochemistry that I wouldn't necessarily find in the database because of incorrect annotations or missing annotations. And once I have that information in my network, I can simulate my model like I showed you a couple slides ago. And by doing that, I get some sort of prediction for what's going on in the network. And then what I can do is evaluate those predictions. I can look at data that actually came from a lab that some biologist measured, and I can compare it to what my model predicts. And then based on those predictions and based on what literature is available and what experiments I've done, I can update my model. I can continue refining it. And this is actually a cyclical process. A model's never really finished. We're constantly updating models and making sure that they correspond to what we know. So now that I've shown you the process of model building, let's actually get to the model that I built. And the process is one part. The other part is what organism do I use. And in this case, I've already mentioned its name. It's called Methanococcus mirapollutus. It's what I might call a methanogen because it generates methane, which most of us know as natural gas, at least the largest component of natural gas. And this one does it from carbon dioxide and hydrogen. It's what I might call a hydrogenotrophic methanogen, hydrogen-eating methanogen. And the way this works is basically shown on the right. I don't expect you to memorize this entire reaction scheme. The important part is that I'm starting with carbon dioxide here, and I'm reducing it using hydrogen here, and I get methane. And these are what those little cells look like. You would find them in, for instance, a salt marsh. They like salty environments. And they're anaerobic. They do not like oxygen. If you expose them to oxygen, they die very quickly. So why is methane important? Well, methane is an energy commodity. We can use it to power various things. A lot of us have gas stoves at home, for instance. And we actually have quite a bit of methane. These particular graphs are from a report in 2014. And what that report suggested was that we have enough methane to last more than 100 years at our current usage rate. So quite a bit of methane. And you may notice this graph on the right is a little bit out of date because I doubt the gas price is really up here. I'm guessing it's more down here at the moment. But it'll bounce back. Methane tends to be pretty cheap, though. And in 2010 in particular, there was a wide gap on a gallons of gasoline equivalent, which is GGE that you're seeing right here. It was pretty cheap in comparison with gasoline. So methane is cheap, and there's a lot of it. But we have to be a little bit careful with methane because it's also a greenhouse gas. It's about 20 times more potent than carbon dioxide. So rather than looking at making methane, what if I could take something that made methane and I could use it? If I could turn it around so it oxidized methane rather than creating it. Well, there are organisms that do oxidize methane, and these are called methanotrophs, methane eaters. And I'm actually showing one of those here on the right. This red organism here is eating methane, and it's producing carbon dioxide. It's basically doing the reverse of what I just showed you. And it's partnering with these green cells here, these bacterial partners. And the pathway that I see in this organism is actually really similar to what I showed you a couple slides ago. Now, again, I did not expect you to memorize that pathway, but if you had, you might notice this is basically the exact same pathway, it's just in reverse. So many of the steps are exactly the same. They have the same enzymes, the same basic genes. And what I was interested in asking was, well, could I take methane up here, and instead of going to carbon dioxide, could I go off here and make methanol? Now, why methanol? Well, methanol is a liquid. And liquids tie more into our fuel economy. If I have methanol, I can synthesize that to butanol. And if I have butanol, I basically have a liquid fuel. So could I take my organism, Methanococcus mirapollutus, and could I re-engineer it so that it went backwards, so that it went from methane and made methanol? So to start doing that, what I did is I created my metabolic model. And the reason I created the model is a good guide for what to do in the lab. Doing things in the lab takes a long time. It can take weeks or months to do a particular experiment that takes me a few seconds to do with a model. So a model is a nice guide for how to do these engineering processes. So my model, which is called IMR 540, the MR is for me, Matt Richards. The 540 are the number of genes in my model. And what I'm showing here is a comparison with a couple of models that some other members of the lab made. Fun fact about those other members, you may notice their names are also starting with M. They're actually both Matts. Every single Matt who's ever passed through the Price Lab has created a model of a methanogen. that is a requirement. So going forward, I have Nathan's assurance he will either hire no more mats or we're going to get some more methanogen projects going. I believe that's right. Anyway, so my methanogen compared to these others, the details I want you to notice here, because there's a lot of information, this is the genome size of the other methanogens. Mine is less than half their size. It's a less complex organism. My organism basically eats carbon dioxide and hydrogen. These other methanogens have a much wider range. They can eat a number of other different things. But otherwise, a lot of my numbers are very on par, if a little bit smaller. And I cover more of the genome because I have less genome to cover. Now, in evaluating this model, I want to really point out the importance of that manual curation step that I don't think I've emphasized enough to this point. So this is the pathway I see in my model. And again, I don't expect you to memorize the entire pathway. Instead, pay attention to the red boxes that I'm going to put. So up here, for instance, there's carbon dioxide coming in. And down here, there's methane going out. And I didn't put a box around it, but right here is hydrogen. And that'll come in a little bit later. So I'm taking carbon dioxide and reducing it to methane, just like I showed in the first slide for this organism. Well, why am I doing that? If I look at the reaction scheme, what I notice is down here, this step, MTR, it basically pumps 2 sodium ions outside of the membrane. And that's really important because those sodium ions then get pumped back into the membrane, and it creates ATP, which you may have remembered is the main energy source for the cell. I create ATP, my cell gets energy, and then my cell is able to do things like grow or stay alive, which it tends to like to do. So I'm creating ATP, and one important step that I've also pointed out there with an arrow to emphasize its importance is electron bifurcation. Now what that basically means is that I have electrons coming in here from hydrogen, and they're split, they're bifurcated. And the reason this is because here at the bottom, I have this compound right here. It's a heterodisulfide. And what I need to do with that compound is I need to split it back up into these 2 compounds right here, these 2 coenzymes. And that requires electrons. But I also need to somehow create this molecule right here. This is ferredoxin. It's reduced ferredoxin. Reduced meaning that it has electrons that aren't in oxidized ferredoxin. So I need to also donate electrons to that reduction process. And instead of doing it separately, I do it all at the same time. So these electrons from hydrogen go to reducing these 2 cofactors, but it also reduces ferredoxin, which then comes up here and completes the cycle. It's actually a cyclical process, something we call the Wolff cycle. And Wolff is basically the father of a lot of these methanogenic processes. So the cycle is named for him. What you also might notice is that there's another reaction up here. This is EHA or EHB. It's what I call a hydrogenase. And it also is actually capable of reducing ferredoxin right here by using sodium ions. And a lot of other methanogens actually have hydrogenases that look a lot like this, some of them named EHA or EHB, some called ECH. And they actually use these hydrogenases as main parts of the pathway to reduce ferredoxin. And when I look at an annotation database, what I would see is that this is a major pathway. This is predicted to really be at the core of methanogenesis. But in Methanococcus, this is actually just a minor pathway. It's what I'd call an anaplerotic source of electrons. It fills in the little bits of reduced ferredoxin that might leak out of the process because it's not 100% efficient, but it doesn't take a main role like the rest of these reactions do. So by going through the manual curation process, and really I'd like to emphasize the role of the fantastic microbiology people I worked with at UW, particularly John Lay. They were really instrumental in helping me look through the literature and determine all of the steps that really go on in this organism rather than just taking what's in the database. So I really want to emphasize how important that manual process was, really not just for this process, a lot of others that I won't have time to get to today, but this one being at the center of it all. Now, I've gone into the kind of qualitative details of the model a little bit here. I want to get back to what the model can do on more of a broad scope from a quantitative point of view. And one thing I want to ask is, how good is my model? How well does it replicate what I get in the lab? But one way to do that is to ask the question of when I simulate my model and I simulate how fast it can grow, how much it can grow from a certain amount of substrate. When I do that, I want to see how well that measures up with what the organism actually does. So what I did is I grew this organism in a reaction vessel, a chemostat, and I measured these points you see that are red X's. So this on the x-axis, if you can't see, this is methane evolution rate. It's how much methane is coming out of the system. And then growth yield, which is basically how many cells are being made per methane. And when I predicted these same points with my model, I got the blue circles. And what you'll notice is that those blue circles tend to fall within the 95% confidence interval of my red X's, the confidence interval, or the bars that you're seeing there. So my model did a really nice job of replicating these data over a range of different methane secretion rates. I also can measure how good my model is by looking at knockout validation. Now what this means is that some biochemist or biologist has gone in and taken a certain gene and asked the question of what happens if I knock out this gene or these groups of genes. Can the organism still grow without those genes? And then I can predict the same knockouts in my model. And I did that. I was able to grab 30 different conditions from the literature. And what I found was that my model predicted 27 of those correctly, or 90% accuracy. Now, accuracy is one way to measure how good the model is. But actually, I'd like to spend a little bit more time talking about the second number, MCC. And that stands for the Matthews Correlation Coefficient, which I really like for two reasons. One of them is obviously its name is fantastic. I'm partial to that. But also, the Matthews Correlation Coefficient goes deeper into the data. It asks how many true positives and true negatives do I have? How many false positives and negatives? It weighs it much more rigorously. And then the coefficient goes from -1 to 1, where -1 is perfect negative correlation, +1 is perfect positive correlation, and 0 is zero correlation. So the closer I am to 1, in this case, the better. And I get 0.67. Now to give you a reference for how good that is and why I'm really pleased with it, this is the same analysis that was done for yeast. This is from a member of the Price Lab, Ben Hebner, fantastic mentor. And he did this for a number of different yeast models. Now yeast, or Saccharomyces cerevisiae, is among the best studied microorganism there is. And the models are, I would say either they or the models of E. coli are basically the highest quality models we have at this point. And the best Matthews correlation coefficients we get are down in this corner. They're about 0.75, though a lot of the data sets he tested on fall below that threshold. So in comparison, 0.67 is really, really strong. So my model is high quality. It performs really well in these different data sets. So now that I've tested the quality of my model, I want to go back and start using it. And one way to use it is by looking at genes that I didn't necessarily know from literature. So I've pointed out that manual curation is important because I might catch things that an automated database wouldn't catch. But on the flip side, there are things that we just haven't studied in the lab so far, that no biologist has gone in and specifically asked a question about yet. And one of those things in my model was glycine synthesis. So glycine, it's right down here, it's an amino acid. We think it's essential for growing this organism. It needs to produce it somehow. And this is the way I think it produces it. And my model predicts this gene right here is the last step in glycine synthesis. So I predict that if I knock out this gene and I don't give my organism any glycine, that I would expect that it would not be able to survive. And this wasn't something I found in the literature. This is something that I predicted based on my automated annotation. And right now, I wish I could tell you I had completely conclusive results on this, but I'm still running the experiment. I have constructed this mutant, and right now I'm doing experiments to grow it with and without glycine. So I can tell you the results are promising, but right now I'm still trying to determine exactly what I got. But it's looking promising that I may have determined the gene for glycine synthesis. So that's one cool thing I can do with my model, is I can look through and start asking questions of, well, where can I expand my knowledge of this organism's metabolism? And that's really important with regards to the idea of metabolic engineering, because if I want to learn all of the things this organism does, my model can help me do that. But I know you've all been waiting to see about this whole converting methane to methanol problem that I mentioned at the start, so I'll get right to that. Now, you may have noticed in this previous reaction network that there's no methanol in here. My organism can't even eat methanol, let alone produce it. So the first challenge when using my model was to get my organism to eat methanol. And actually that's a pretty easy challenge. A lot of methanogens do eat methanol, and so by putting in that same gene, and it's right down here, it's a methanol methyltransferase, or MTA. And all that does is it takes methanol and it puts it into my existing pathway. Now you may have noticed that all of these reactions right here are now in orange or brown or whatever that looks like to you. Those reactions are now relegated to minor status. They're really not doing nearly as much as the reactions in black. And in fact, this step right here that I mentioned was so important for driving sodium outside the membrane, well, now it's going in. Sodium ions are pulling in. So how do I make ATP in this case? Well, actually, that EHA or EHB that I mentioned before now becomes the main sodium pump. So what it does is it takes reduced ferredoxin from my heterodisulfide reductase, and it uses that to drive out sodium ions that then come back here into ATP synthesis and make ATP. And I can do that because this step right here, this carbon dioxide to formylmethanofuran step, is no longer a major part of my pathway, and in fact, it's now going backwards a little bit. So this is how I go from methanol to methane, and that's really the simple part of the process. The more difficult part is how to go backwards. And the reason that's difficult is because now I have to take this step here, this heterodisulfide reductase that I've talked so much about, and I have to make it go in reverse. And so what that ends up looking like is now that HDR is creating hydrogen, and it's using reduced ferredoxin. But the problem is I also have my EHA using reduced ferredoxin. So I need some way to get that compound. Well, as you might have guessed by the fact that it's red, I was able to accomplish this by putting in a different enzyme, a different enzymatic reaction. I call it FHOR, or ferredoxin hydrogen oxidoreductase. And what it does is it takes the hydrogen that's produced by HDR running in reverse, and it uses that to reduce ferredoxin. And this completes the pathway stoichiometrically, and it allows me to take methane over here and convert it to methanol using the reactions you see here, basically a simplified version of the network. Now I said that completed the pathway stoichiometrically, but what I haven't really addressed yet, and I'm going to touch shortly upon, is energetic feasibility. My organism doesn't want to be working uphill the entire time. It doesn't want to be producing energy. It would rather be working downhill, basically using energy. And if you'll recall, when I first introduced this idea of eating methane, I showed the archaea, the red cells, with the green cells. They were working together. Well, what those green cells were doing is they were doing a reduction. They were actually taking sulfate here and converting it to sulfide. And what that reaction pathway does is it basically produces negative free energy, the kind that helps the cell be able to grow favorably. Cells want to produce negative overall free energy, not positive. And so by pairing with the bacteria that's doing one of these reduction pathways, I'm able to have an energetically feasible process. And by testing these in my model, what I found was that nitrate reduction was actually able to complete the pathway. If I allowed my organism to reduce nitrate, assuming I could get those enzymes in, then I could carry out this reaction here, and it was able to drive free energy to 0 at standard concentrations of 1 millimolar for each reactant. And by manipulating concentrations, I could feasibly even go even lower. So what I've done is I've been able to complete both the stoichiometric and energetic pathways here. And this isn't something yet that we've been able to do in the lab. We're still working on getting methanol to go to methane. When I say we, I mean me and my collaborators at the University of Washington. But now I have kind of a roadmap for a possible way to do this, and I can continue working with my model to get more ways that I might be able to go backwards. So to sum up this section, I want to emphasize again how important the manual curation was, not just for what I showed you in the bifurcation, but for a lot of other pathways I just don't have time to show you today, like coenzyme biosynthesis. And this is what takes my model and it really makes it specific to my organism. But the automated curation also has its merits, mainly that it can suggest different things, make different predictions that I wouldn't have known about from literature. And ultimately, using my model, I was able to predict a pathway from methane to methanol. And so we can use that as a guide as my collaborators and I are working to actually make this a reality. So I want to get to the last part of my talk now. It's going to be a little bit shorter than the previous part, which actually combined 2 chapters of the dissertation. And in this part, I want to talk more about a method that I've developed that allows me to take a high-quality model, like the one I just talked about, and propagate that to a closely related organism. Now, I showed this graphic before, but what I didn't really mention is that this process of making a high-quality model takes something on the average of about a year, usually a year plus, I would say. And so that's a long time to put into one organism model, and if I want to learn a lot about, say, this entire tree of organisms, particularly these ones in red, then it would make sense to have some sort of way to cut down on that time. For instance, if I made my current model, the one that took me a year or so, and I wanted to make a model of a closely related organism, like another methanogen, couldn't I take some of my model and pull some of what I've pulled out of the literature into that new model and use that kind of as a first draft, as a starting point? So I'm not just starting from automated curation, I'm incorporating some of what I pulled from literature. And I've created this method with a lot of help from a fantastic undergrad, Brendan King, who works with me out in Seattle. And what this method does is I feed in the high-quality model that I created earlier and its genome, and the genome of an organism that's very close to it. I also need to feed in the growth conditions, the media for organism B, so another argument in favor of having a database of these conditions readily available so I don't have to go scouring the literature. Now this model, of course, is the one that I've just spent the last 20 minutes talking about. It's the Maripolutis model. The features of that model. I'm not going to spend too much time on this, but there are basically over 500 genes for me to compare to here. Now when I say compare to those genes, what I'm talking about is what I would call a proteome comparison. So in the proteome comparison, what I'm doing is I'm taking my 2 genomes and I'm feeding those in to this part of the process, and what I'm really doing is I'm comparing those genes to see where do they line up. So if this is my model's genome, I see that the next genome I'm propagating to shares this gene. Gene B1 is the same as GA1, or similar enough. So I would infer then that the reaction that is associated with gene A1 right here is also in reaction network B and associated with gene B1, and so on and so forth for the rest of the network. Now this will pull a subset of my genome scale network. It won't pull the whole thing. So I'm going to have some gaps here in red, but I would expect that because my organisms aren't exactly the same. They're going to have some differences. So now I have what I would call translated model B. This is the mapping of my organism, my new organism onto my manual model. Well, I know those organisms aren't exactly the same, so what I can do is I can feed genome B into my automated database, K-base in this case, and I can pull out the automated reconstruction, or recon B. I can also create the reaction probabilities object. And to remind you about this is using the algorithm from Matt Benedict, my former lab mate, which goes into a metabolic network gap like this and fills it with probabilities. What I'm doing in this step isn't actually filling those gaps, I'm calculating those probabilities so I can see what reactions in the database don't necessarily pop up in the reconstruction or the translation, but are really likely to be there. And by doing that and setting a tunable threshold, what I'm able to do is I can combine those really likely reactions with my translated model and with my automated reconstruction of a model. And doing that, I get what I call supermodel B. Actually clamoring from some of my lab mates to put a picture of a supermodel here, but I decided against it. You can be the judge of whether or not that was a good decision. Now that supermodel has all sorts of reactions from these different sources in it, but I don't necessarily have the assurance that it grows on my new medium. In fact, if organism B grows on a sufficiently different set of growth conditions, I might have to do a fair amount of gap filling to make sure it can grow. So that's the next step, is I gap fill my model based on those growth conditions. And then the final step is that now I have a whole bunch of reactions, but I haven't actually removed anything yet. I've sort of pulled genes from different sources and I've filled in gaps. Well, there's going to be a lot of information that's sort of redundant in my network, right? I don't have genes for certain reactions, but there's a whole bunch of different ways to get from point A to point metabolite 1 to metabolite 2. So I want to pull out these gap-filling reactions, basically things that don't have genes, so I don't really have anything in the way of genetic evidence for them, but that also don't fulfill an essential niche in the model that I don't need in order for growth. And that gives me my final morph, or morphed model B. Now, to actually demonstrate that, what I've done is I took my model, so Methanococcus mariplutis right here. This is a phylogenetic tree of the different methanogens that was constructed about 15 years ago at this point. And in looking at this tree, I can see that Methanocaulococcus right here is actually a decently close relative of Methanococcus. And I picked it in part because there's quite a bit of literature on it, and a lot of the information that went into the mariplutis model actually was originally discovered in Methanocaulococcus unacii. So if I were constructing a model of unacii, it would be nice to pull a lot of that literature in without having to go and manually find it again and pull it in. I also chose Methanosarcina barkeri. The Methanosarcina are quite distantly related to the Methanococcus, even though all of these organisms produce methane and can do so from carbon dioxide and hydrogen. They're actually quite different. And you may have noticed one of the big differences is that Methanosarcina barkeri has a much larger genome than either of these two. Methanococcus is around 1,700 genes, and unacii is around 1,800. Barkeri is more in the range of 4,000. And so when I do this model morphing, what I find for Methanocaulococcus unacii is that my morphing process takes what I would have gotten in the automated model right here for genes, and by taking genes from my model, I actually get more genes. And some of this is going to be because there's disparate genes in the two different sources, but a lot of the reason that I get this increase in genes is because I'm getting quite an overlap between my model and this new organism. And there is some increase in the number of metabolites, and that's because there's going to be things in my manual model that don't appear in the automated model, so they're going to get added to that. But I don't have a huge increase in reactions, and that's because in the last step, I'm pruning a lot of those things out and trimming the non-essential parts. Comparing this to Methanosarcina barkeri, I see that barkeri ends up with quite a few more genes, and I would expect this for two reasons. One is that it just has a larger genome. So if I take a larger genome and I feed it in, I expect to see more genes in the final product. There's also just going to be a lot less overlap between this organism and my starting organism, so I'm expecting to see a larger influence of the genes from K-base in the final model, the automated model. And similarly to the other one, I see an increase in metabolites, but not a huge increase in reactions. Now these are really just they're general numbers. They give you some idea of what's going on, but to learn more about what's going on, I can actually look at where the reactions in the final morphs are coming from. So this green circle over here is the final product I get, and the other two circles are the source materials, the blue one being my model that I've talked extensively about, and the yellow one being the automated reconstruction. What I can see is a few things. First of all, these reactions here in the middle are the conserved core. They're the things that pop up in my automated model and in my manual model. So those end up in my morph. But then comparing the things that don't end up in that conserved core, what I find is I take about 2.5 times more reactions from my manual model than I do from the K-base model. And in doing so, I'm actually able to eliminate about the same number of reactions from K-base. So I've trimmed out a lot of this information that I wouldn't necessarily have genetic evidence for, and I've replaced it with a lot of information I do have genetic evidence for from lining up my two organisms. So by doing this, I've sort of gotten a head start on a lot of the manual curation, and kind of given whoever takes this model and turns it into a full manual model a nice head start on that. Now comparing this with what I see for Methanosarcina barkeri, I still see this really nice core here of about 350 reactions, and I actually do still take quite a few reactions from my original model. But barkeri has a lot more going on. It's a more complex organism, and it's further from my organism. So instead of taking more information from the source model, it pulls many more reactions from the K-base model, about 200 of them. So even though I'm still able to eliminate these about 150 reactions from K-base that don't necessarily have any genetic evidence, I do take a large portion of the K-base reactions to create my final model. So it seems like there's some sort of influence just in terms of both genome size, I would say, and distance from my organism. You can also notice that these red percentages here, it's a lot lower. If you look back on Yanashii, about 80% of the reactions I'm taking from my original model have genes versus about 70% on the case of Barkeri. And the last thing I want to look at for these two morphs is the genes. Where are the genes coming from? And where I see them coming from is in the case of Yanashii, even though I have a smaller genome, a significantly smaller genome than Barkeri, I'm actually not only taking a higher percentage of genes from the manual model, I'm taking more genes total, about 50 more genes. So about 60% of the genes in my final model are from the Maripaludis source model versus only about 40% of the genes in my Barkeri model. And instead I'm seeing the Barkeri model gets most of its genes from the K-base. But in both cases I'm actually seeing really poor overlap between the K-base and my manual model. So I wanted to investigate what was actually going on here, if there was an artifact in the process. And to do that, what I did is I morphed back to Maripaludis. So I'm basically taking the same organism and creating a morphed model for it from my manual model. And what I see is that instead of just keeping about the same number of genes, I get a huge increase in the number of genes. And there are two reasons this might be happening. First of all, I might be adding a lot of reactions from K-base and bringing the genes there. Or I might be having the same reactions but disagreements in the genes. So to examine that, I looked again at the reactions and I found what I might expect from morphing to the same organism. I took very few reactions that were just K-base. There's a big core of conserved reactions, and most of the reactions come from my model. So this final morph does what I expect it to in terms of reactions, but the big difference comes in terms of genes. So I actually see I take the most genes from my model, as I would expect. It's the same organism. But there's really, really poor overlap between my manual model and K-base. So what this suggests to me is the idea that if I'm taking an organism like mine that's sort of off the beaten path, it's not your typical E. coli that eats glucose. If I take something like that and I go into an annotation database that's based off a lot of these more generic organisms, I might not be able to do as good a job of pulling these same genes that I would get from actual biochemical characterization. There's a lot of information that I can miss, and there's a lot of misinformation that I could be layering on here. So I think this demonstrates why I think this morphing method has a lot of potential, is if I'm going to go out and construct a model of something that's really close to Maripaludis, it makes a lot of sense for me to start from a method like this, where I can pull a lot of the information that I've gone through and gotten from literature, and sort of have a guide that can point to me where I might want to be skeptical about annotations from K-base versus the ones from literature. So to wrap this section up, I want to emphasize again that there seems to be some sort of trend, at least looking at Yanashii and Barkeri, and there's actually another morphing I did for an organism that's in between that sort of suggests a trend with the phylogenetic distance compared with what the final network looks like and what I pull from my model. But ultimately, I think the bigger takeaway for me from this section is the idea that we have to be careful about what we take from automated curation, because there's no guarantee that what we take is going to be real information rather than misinformation, and that they can really diverge from one another. So with that, I want to go into my final conclusions for this talk. And the first one is to, again, emphasize the caution that we need to take with generalized approaches. Now this graph I know can be a little bit difficult to interpret, so I'd like to point out that B and C, right here, B and C, are just zoomed in version of A. This is the same graph, it's zoomed in so you can see more detail. And what you're seeing here, every single data point is a metabolic model constructed by somebody. And these metabolic models are mapped out using a multi-component analysis. It's basically grouping the different models based on how similar they are to one another, based on what genes and metabolites and reactions pop up. And what you see for the most part is that almost everything clusters really closely together. There's a couple of things that pop out, an algae model, a corn model, but all these microorganisms, which you can see more clearly in here, there's a lot of dots, and they're really different from each other. You've got Geobacter metalloreducens, which is a soil organism, and I wouldn't expect it to look that much necessarily like Mycobacterium tuberculosis, which is right here. But all these things look really similar. And so I think the idea I'm trying to emphasize here is that if we go into these automated processes and rely too heavily on them, what we get is a lot of models that look exactly the same or very similar. So we're robbing ourselves of the chance to discover a lot of microbial diversity. And if we want to be able to employ metabolic engineering processes and develop industrial biotechnology based on it, it's important to be able to discover these bits of diversity that set organisms apart. And so in moving forward, it's really important, even with my own resources like media database, I need to recognize that even though those resources are really valuable and they can help me speed up processes and bring information together, I have to be very willing to go outside those sources and check on the information and supplement it and make sure that I'm pulling the manual information to really check on what I've done through the automated method. And that sort of combined approach, I think, is how we're really going to make headway on answering these questions of how microbes function and be able to leverage their metabolisms to do some really cool things in industrial biotech. With that, I want to finish up by thanking all of you for being here today, committee, everybody here, and of course the entire group back at ISD. This is our latest lab picture. There's one lab mate missing, and so we've graciously filled her place with the Dalek. All the folks in orange, thank you so much to them. They're my co-authors or collaborators. And a special thanks, I had to group them together just for space, but the Lay Lab at the University of Washington, a product of the Methanogen community, John Lay and his group were nice enough to let me come in and poke and prod at actual organisms and do all of this work. And I think if I had advice for somebody putting together a model in the future, it would be make sure that you're aligned with a really, really good biologist or biochemist because it makes the entire process much more painless. So with that, I'd like to open it up to any questions from the audience, and thank you for listening.
All right, audience questions first. All right, if not, then we'll excuse everyone and we'll go into closed session.